So as you know, Simple Western is, these are very powerful instruments with the very, I would say, very diverse set of applications. And again, this SPA is going to be, we're going to talk about the basics, just the beginning. There are a lot of things that you can do with these instruments. We have this small box that can take up to 24 different samples where you can really test a lot of stuff in a minimum time. We also have the big box, which is more like a high throughput. We have the Salisu and the PeggySu, where we can you can test more samples, up to 96 different samples, which is huge. So the point of this instrument is really to streamline your process and, to be able to get results fast, because this is a very important concept in these days. So in terms of the internal mechanisms, and I'm sure again that some of you know, it all starts with the instrument essentially made of the capillaries, the capillary electrophoresis. So we have the separation and stacking matrix that is being aspirated. Then we have the sample aspiration. On your plates, I don't know if you know, but you put 3 microliter, but the instrument essentially takes much less than that. It's probably about estimated around 40 to 50 nanoliter of sample. And then the samples are running, and then there's a separation by size, and then the instrument immobilizes the proteins onto the capillary wall, and then the probing process happens in the measurement. So that's essentially the internal mechanism of the instrument, for those of you that don't know. So as I said, there are 2 modes of detection on the JES instrument. Specifically on the JES instrument is the chemiluminescence channel, and we have the fluorescence channel, the NIR and the IR. It's a very similar concept. It's just that one of those, you're going to have to pipette your luminol peroxide, and for the fluorescence, you won't need your luminol peroxide. The outcome of this is an electropherogram. The electropherogram is your data where you could calculate the area under the curve. The area under the curve is what matters. Is essential for in a regular Western lot, when you have your film, then you now go to image A or whatever, and then you kind of circle the bands and stuff like that, and things might go off and not be as accurate. Here the accuracy is very high. You'll get an electropherogram that this is actually your data, and it's going to provide you a lot of insights into your protein. So in terms of if you have PTM, post-translational modifications, if there are several isoforms, there's a lot of value to these electropherograms in terms of the scientific output that they bring. So I'd like to talk a little bit about the workflow. The workflow in the lab should take about 45 minutes. It doesn't really take long. But it starts with our easy standard pack. Our easy standard pack has 3 different types of packs. There's the 2 to 40 molecular KD. We have the 12 to 230 KD, and we have the 66 to 440 KD. And as you can see on the left side, you'll have 3 different vials. One of them is going to be the DTT, where you pipette 40 microliter of deionized water to make a 400 millimolar solution. Then you have your fluorescence master mix. This is 5 times fluorescent master mix. You'll see this, the color is pink, where you will add 20 microliter of your 10 times sample buffer and 20 microliter of your DTT. You're going to mix it well, and these are going to be your standards. Then you have your biotinylated ladder, where you add 20 microliter of water. Essentially, the standards, these master mixers, going to be incorporated into your samples in a ratio of 1 to 5. So you'll have your 4 parts is going to be the lysate, and 1 part is going to be the fluorescent master mix. What I like to do is I calculate, how much volume do I need? I divide it by 4 and just add that number to my sample from that pink solution, that fluorescent 5X master mix. And then the next step would be to boil your samples, to denature your samples. Now, there are different samples. Different samples require different treatments. Sometimes 95 degrees for 5 minutes is great. If you have a transmembranous protein, you might want to consider not boil it or boil it less on a low temperature. So there are a lot of considerations, but this is part of your assay development, part of the process that you go through to figure out what are my best conditions for my experiment to get the best and high quality data. So in terms of the plate itself, you'll pipette your samples, you'll pipette your ladder. The samples essentially, as I said, is about 3 microliter into these wells. I would remind you, or if you know and if you don't, to avoid bubbles. And especially in row A, when you have bubbles, you might lose the data. So it's really important to make sure that you don't have bubbles. Then you will load your antibody diluent, you will load your primary antibody and your secondary antibody. And if you run a chemiluminescence, then you'll have to load luminol peroxide. This is 15 microliter. You will also load your wash buffer, okay? And it is important, and again, for the reason of, because of the bubbles, it is really, really critical for you to spin it down in a centrifuge that has an adapter for plates. For like, I would say 1000 G for 5 to 10 minutes. It's really up to you. Usually we recommend 10 minutes, but 5 minutes should be sufficient as well. Please remember that when you spin your plates down, that the centrifuge is not cooled down, because what can happen is that, you know, under like our matrices can aggregate. So you really want to make sure that you keep the plate in room temperature. So it's really important. Then after you spin down your plate, you'll remove the foil. So you'll have the top foil, which you'll remove at the beginning before you load your plate. And then you have the bottom foil, which you're going to remove just before loading the plate onto the instrument. The reason why we recommend doing that is because we want to avoid evaporation of the different reagents that are here. So, okay, so when you peel off that foil, that bottom foil, you might generate some bubbles. So what I personally do is that I take a little, you know, a small tip or a needle, and I just try to pop the bubbles. When you do that, try to not cross-contaminate between the different rows, because different rows contain different materials. Definitely don't introduce, for instance, if you have a bubble in row A, definitely don't use the same tip and go to row C, because in row A you have SDS. So you really want to make sure that you do that. But yeah, popping bubbles can really minimize the chance, because sometimes even if you spin it down, you might see a bubble or two. You can use a needle and it works great. So these are several tips for you when you start working. After that, you want to, you know, select your assay. So on the instrument, you'll see that screen. When you'll choose new assay. Then you'll select chemiluminescence or fluorescence. You have the ability to select replex, stellar, protein normalization, the size range, and whether you use a cartridge that is 25 or 13. And then you click OK, and this screen is going to appear. In this screen, I personally, listen, you don't have to fill up your, essentially your samples and what type of primary or secondaries you added in. But I always like to do it before I'm starting my experiment, because knowing myself, and I think a lot of people share this type of behaviors, we never do that and we don't remember what we ran. So I highly recommend always, you know, fill it up before you start your experiment. But again, it's up to you. There are a couple of interesting values here that it's good for you to be aware of, because for some of those things, you can change those to adapt the run to your needs. And again, as part of your assay development, and the first thing you want to consider is the stacking metrics. The stacking matrix, I don't have it here open, but if you click on the stacking matrix, you'll be able to see a value that is in seconds. It's essentially saying how many seconds does the instrument how much of the stacking matrix the instrument takes. So this is something that could be beneficial to play with. Maybe not right at the beginning, but if you want to have a better separation between peaks, this is something to consider. The next thing that I think that is the most important that can really help you is the primary antibody time. The default is 30 minutes, but for some antibodies, and I've seen it myself, 90 minutes will really improve your results. So just be aware that you can change your experimental conditions in this screen as well before you run your experiment. Before you run your experiment, let's say you made a mistake and accidentally in row A, you skipped some samples, you put the wrong samples. Oh my God, do I need to throw everything out? The answer is no. Sometimes you're able to just drag A to row F and then essentially start, moving your samples here to row F, and the instrument is going to take it from row F. So just know that at the beginning, before you start running your experiment, you can drag those things around, to row F or to any other row. So just be aware of that if something happened, it's not the end of the world. You might change it. There are other protocols where we use the whole plate where we cannot actually do it. But if your experiment is very simple, then you might be able to drag A to another row. So, okay. And then in terms of the high dynamic range, we always recommend leaving the high dynamic range is always the best. It's essentially taking all the exposures together, which is what we recommend. Okie doke. So let's see if I forget anything. Okay, so the next slide that I want to talk about is your experimental design. So again, I think that a bad antibody will definitely waste your time, your money. So you really, the first step that we always recommend starting with is to scan multiple different antibodies, and then see what works for you, because most antibodies will work in this system, but you might have an antibody that might not work, and then you will struggle for a longer period of time. And so if you can, order for, let's say, for the same target, 2 to 3 different antibodies or 4 antibodies, and then scan several of those and see which one of those has the best peak, which one of those doesn't have background. Those things will benefit you if you do them early on. Online, we have an antibody database that you can access where you have antibodies that were validated with Simple Western. There are more than 3,000 different antibodies on this database. So some of it might help you kind of figure out what are the conditions, the experimental conditions you'd like to you want to move on with or start with. So part of the topic of this presentation is really to get you started, right? Just to get you started to what are the do's and don'ts, let's say, at the beginning. So one of the things that we have to watch from is burnt out signal. If you have a burnt out signal, it might look like that. So you see that blue button here with the squares. If you click that one, then you might be, you will have like a checklist, and then you might check the all exposures. And when you check the all exposures, you might be able to see, the different exposures. Now, if you see something like that has this huge drops, that is a sign that your signal is way beyond the linear range, and it's burning the camera, and there's nothing you cannot quantify with it. So it's really important to make sure that you're not using your signal is not burned. One of the indicators that I like using for chemiluminescence, for instance, if the peak is higher than 300,000, then that's an indicator that you see here it's 1,200,000. It's 1,100,000 to 200 or something like that, right? Plus minus. So this is an indication. So this signal is too much. So what we recommend doing in that case is really diluting your sample. Sometimes antibodies are sticky, and really it depends also on your sample and what you use. They might stick to a lot of stuff and they might create background. So the same button, if you click on it, you're able to turn on the background and see, if you have background. And if you do have background, then you might want to consider dilute your sample or antibody. So these are the things that you can just do when you see your results. There's a couple of things that you can do to see where you are. A lot of people ask, they see a peak at, let's say, 60 kD, and they're like, okay, is this my protein? Is it not my protein? Is it something else? What's happening here? So what I always recommend is, if you have a knockout model in the lab, or if you have a siRNA that you can infect your cells, I don't know, 293T if you're using, or something like that, or another cell line. Use those methods of overexpression or knocking down, because that will be able to help you validate whether the peak, the beautiful peak that you're seeing in 67 is actually your protein that that it's in 59. Okay, so I think it gives a lot of people, using this type of methods gives a lot of people the confidence that this is what they're looking at. Similarly, when you look at the regular Western blot, when you see a band, right, like you want to know, is this really my protein? Is it not? Is it just an artifact? What is it? So peak validation is the way to go. So I highly recommend using knockout or overexpression to make sure that this is really your peak. So we're not really going to discuss assay development in details here, but I would say that when you develop your assay, so as I said before, the first step would be to avoid bad antibodies and potentially screening multiple antibodies. And then the next step would be to select the right antibody concentration. You really want to make sure that your antibody, your primary antibody, is really in abundance. This is really critical, because if your primary antibody is not in abundance, then you might introduce variability to your assay. Any small, little, tiny pipetting mistake can introduce variability. So you really want to make sure that you have enough of it. So this is the next thing you want to do. So the way to do it, and the way I like to do it, is that I plot the antibody dilution, so different dilutions versus or against the area under the curve. And then I pick that green area. So I see which concentration is starting to saturate my signal. And this is concentration. So in that case, it would be 1 to 40, right? So I'm going to pick 1 to 40 in that case. But again, every assay is a little different, but you really want to make sure that you are starting to saturate. You're not completely saturating, because if you are saturating completely, then you might see a lot of background. But this is really the sweet spot when your signal is starting to saturate. So you have a lot of antibody and enough. So this is the second part, to really make sure that you have enough primary antibody in your assay. The next part would be to look at your dynamic range, to really make sure that your sample or your signal is within the dynamic range. Dynamic range of assays would be about 4 logs, but you really want to make sure you want to run different concentrations of your sample and plot them against the area under the curve. And you want to select that area that is within that linear part of the graph, because this is the best area where you can actually quantify. So again, same method. I like to plot different concentration of my sample versus the area under the curve, and then I pick that concentrations that are within the linear range. What I also like to do is that if I'm expecting an upregulation of my signal, of my protein, right? So I might select the area, or I might select the concentration, the dilution of my sample that is actually on the lower part of my dynamic range. If let's say if I'm expecting a downregulation, so I might want to check the upper part of that dynamic range. And the reason is to just really make sure that most of your samples or all of your samples are within that dynamic range, within that linear range of your assay. That's really the best way to measure it. We offer a kind of a quick start. It's kind of an idea, you can take 3 different antibody concentrations, 3 different sample concentrations, you add to it 2 more controls. One is no primary, no lysate, and you would just have it all. You might have in a 24 capillaries, you might have 2 different targets. I personally prefer to use 5 different points on my graph so I can really catch those areas, whether it's the saturation point for my antibody or when I see the full dynamic range of my of when I dilute my samples. So I like personally to choose 5, but definitely if you want to do like have a quick start and you're in a hurry and you want to get results, because we can, some of us really, assay development is really important for us and we want to be accurate and everything, but some of us just want to start. We just want to start and we understand that. And so this is why we came up with this 3x3 matrix that can be very helpful. Hold on a second, 1 second. The post-flight checklist. So after we select the, just a second, 1 second. After the quick start, we're going to look into the post-flight checklist. Now, the post-flight checklist is really important because we have this tendency to be like, oh, well, my results, I want to see them, I'm excited. That's awesome. But it's really important to take a step back and look at your data file to really make sure that everything is fine. So the first thing you want to look at is really looking at your standards to make sure that your standards are called correctly. This is a number 1. If the standards are not called correctly, then you can right click and force the standards for another peak or for another area on this graph. So that might happen. It's really rare, but it might happen that your standards are not called correctly, in which you will have to fix it. The next thing you want to look at is your ladder. You want to make sure that your ladder is called correctly and it looks good. If your ladder is not called correctly, and I don't have this picture here, under edit analysis, you're able to cancel some of your ladder or your whole ladder, and that's fine. But you want to be able to look at these things, because if you don't look at these things and you see a pick at 60 and you expect a pick at 85, that might be because your ladder is incorrect or your standards are incorrect. So taking a second to check, taking a second to really making sure that your run file is okay is very critical. And the next thing is really that video. This is the separation video, and this separation video is also giving you an indication of what's happening. So in the bottom, it's inverted, but in the bottom you see your 1 kD, and this is for the 12 to 230 separation module. So the bottom, the strong signal is the 1 kD. The second one is the 29 kD, and the upper one is the 230 kD. So by looking at this video, you can see whether you have bubbles. Sometimes if you see a bubble, it can look like a shooting star. So then you might know, okay, I don't see data there because I have a bubble, and that makes sense. Next time, we should avoid or pipette very carefully. So that gives you a lot of information. The next thing you want to look at is the IV plot, which is here. You want to look at the sigmoidal curve. And then you see this noise on the right side, on the top. That noise essentially signifies that the UV light is turned on. Okay, so this is the post-flight checklist, and it really is important for you to take the time to look at this, because otherwise you might have problems or issues that are not related to your assay, but is something that is more global. Okay, so I'd like to shortly speak about our platinum products. Our platinum products are, we're going to discuss them, I think, in a detailed manner later on our next workshops, but I'd like to just mention them at least. We have the Replex module that allows you to strip your capillary and reprobe, so know that this option exists. This is how it looks like. We have our total protein assay, our TPA, and the TPA essentially requires Replex. So you have your immunoassay, there's the capillaries are stripped from all the antibodies, and then they're being reprobed with the TPA reagents. So the TPA is essentially using chemiluminescence. It does extend the run for 5 hours. I personally like the TPA a lot because the TPA really allows you to normalize your signal to something that is not a housekeeping. Housekeeping can be sometimes very tricky because it might be impacted by your experiment or your treatment. So you really want to, so by using TPA, you really kind of neutralize that, and you're just able to kind of normalize your signal to the TPA. The TPA would look like, essentially, it's like a Comasi. It's going to stain everything. So any small changes in protein expression will not be as significant. So this is why I personally like the TPA a lot. The dynamic range of this assay is between 0.05 to 0.2 mg per ml. We have another product that is called the protein normalization, the PN. And this is essentially, this is not using chemiluminescence. It uses a separate channel, a fluorescent channel. It's essentially a fluorophore that binds the essentially the amines and proteins. So again, the same principle. It binds everywhere on the capillary, and you can normalize the area under that curve. You can normalize the area under your peak against that area of the PN. The difference here that I think PN can be better because for a lot of reasons, but also the TPA is great. But I think the PN has an advantage because it doesn't add more time to your run. But some of you would say, okay, I mean, if I'm doing it overnight, I'm fine. So it doesn't really matter whether it's 5 hours or 3 hours. And then the dynamic range of the protein normalization, the PN, is between 0.2 to 1.2 mg per ml. So it really depends kind of what is your sample concentration. If your sample concentration is low, then I would definitely use the TPA. If your sample concentration is a little higher, I would use the PN. If the sample concentration is higher than 1.2 mg per ml, then we'll recommend you using a housekeeping. Okay, so as you know, on the JESS system, we have several different channels. We have the fluorescence channels and we have the chemiluminescence channel. Usually the chemiluminescence channel is much more sensitive than not much more, but it's a little bit more sensitive than the fluorescent channels. This is why we came up with this stellar assay, and just know that it exists. And again, we'll have a whole session on stellar. So stellar provides essentially the same sensitivity of the chemiluminescence channel.